Thank you everyone for attending the talk today. And I hope you had a fun and productive week here at PEGS. So today I'm going to introduce you to a new service that GENOIS Azenta is about to launch, Sequencing to Synthesis. So let me start with giving you an introduction about the company. GENOIS, among the company that you see listed here, were rebranded recently in 2021 under the Azenta Life Sciences brand. GENOIS brought to the group many years of experience in the multi-omics market. We have solutions that span from genomics, transcriptomics, and proteomics. The many customers that we have had over the years have continued to recognize and appreciate GENOIS' superior data quality, our unparalleled technical support, and very fast turnaround time. Azenta is a global organization. We are present in North America, both in the US and Canada, and we are also present in Europe, Germany, and UK, and in the Asia-Pacific region, in China, Japan, and Singapore. So today I'm going to represent the Azenta Life Science company and brand, but the solutions that I'm going to introduce you today are being offered by our GENOIS brand and company. So GENOIS has many years of experience into reading the genome and the transcriptome, and that is done through our sequencing capabilities, but we also have capabilities into writing genes through our synthetic biology solutions. We have supported and we can help scientists that are working through the entire antibody discovering the development journey, from discovering and preclinical testing all the way to clinical trials and manufacturing. We can help also managing your samples, storing your samples, and with the transportation of those. So today I'm going to introduce you to an innovative antibody discovery workflow that GENOIS is about to launch. As I mentioned before, GENOIS has strong capabilities in the sequencing part of the workflow, and we also have strong capabilities in the DNA synthesis and antibody production. We realized there was an opportunity and a gap into our workflow that could be implemented with computational analysis. So we are now introducing an antibody discovery module in partnerships with OpenEye and Specifica that will help analyze the sequencing data to identify the most optimized leads for gene synthesis. So how does high-throughput sequencing, next-generation sequencing apply to antibody discovery? We have heard about this topic, a lot of discussion during this past week, and here I'm citing some recent work from Specifica that really highlight how the next-generation sequencing can implement the diversity of the antibody population for screening. NGS can really help in two scenarios: when the diversity is dominated by few clones, as well as to expand the clone diversity to identify clones that have similar features like binding on a specific target, but they have different beneficial features like different sequencing liabilities. Reducing the sequencing liabilities eventually will support on the developability of these antibodies in downstream work. NGS can be used both it can be used as a standalone pipeline or in combination with Sanger sequencing. When used in combination with Sanger sequencing, it will provide additional and deep information on a selected population of antibodies. GENOIS has many next-generation sequencing solutions for immunogenomics. We offer a solution for both the analysis of in vivo repertoires and in vitro display. For the analysis of in vivo repertoires, we offer both bulk RNA analysis and single-cell analysis. And for the in vitro display, we can offer amplicon sequencing on short reads Illumina, as well as long reads PacBio. Let me dive a little bit more into our sequencing strategy. So for our bulk RNA analysis, we have developed our proprietary amplification method, and we can accept multiple and different types of starting material. For the single-cell RNA analysis, we use the 10x genomics technology, and we usually suggest using this technology when customers are interested in retaining their heavy and light chain pairing information, and when they want to access frequency analysis at the single-cell level. For our amplicon sequencing for in vitro display, we offer both solutions on the short reading sequencing on the Illumina platform, as well as long read sequencing on the PacBio platform. We suggest to use the PacBio solution when customers are interested in retaining the heavy and light chain pairing information, because the fragmentation of the amplicon in this case is not required. We can sequencing a genus amplicons that are as long as 10,000 base pair on the PacBio platform. As I said before, we have strong experience in the sequencing world, but we also have years and decades of experience in the DNA synthesis market, and we have recently added capabilities for the antibody production. We currently have a full end-to-end solution for antibody DNA synthesis through recombinant antibody production with very fast turnaround time. In fact, our DNA antibody synthesis is one of the fastest on the market. We can start delivering results in as few as 6 days, and we can deliver also large volume of project up to 1,000 constructs without adding any turnaround. In addition to this, the sequencing is fully guaranteed with a quality control performed after the synthesis with Sanger sequencing. For our recombinant antibody production, we have multiple options that our customer can choose upon. We have different types of antibodies. We have different expression systems and expression volumes. We can go up to 1 liter of large expression when needed. And we have developed in-house some expressions vectors for mammalian expression that have been optimized and provide a very robust expression. However, we can also accept customers' vector. Standard QC is performed to ensure that we deliver high concentrated protein with very high quality. We check for aggregation, for purity, and our technical support team will really help customers to go through these entire options and find what are the most reliable for their project. We will provide a project consultation, help with the project design, and provide updates at each step of the project throughout its completion. Here is a case study that there was a pharma company that wanted to partner with Genuis. They had a challenging project where they had never expressed this IgG. There were over 20 human IgG that they wanted to express. It was new, so they didn't really know how much expression they could expect, but they wanted very high concentrated protein in a very fast turnaround time of very high quality. So Genuis was able to perform this project very successfully. We had a very customized project in this case. So the VH and VL cloning was expressed in a customized vector, and we also had to use customer buffer. The results were highly concentrated proteins, as you can see here. We had a concentration that ranged between 13 to 14 milligrams per ml. We also had very high purity proteins. As you can see in the last column of the table, the purity was above 89% for all of them. And we have very low endotoxin levels, which is also indicated here. For most of them, it was near zero. The project was delivered in record time in about 3 to 4 weeks. With that, I would like to dive a little bit more into the antibody discovering module that we are now adding into our workflow. So the bioinformatic pipelines that allows for the prioritization of the optimized leads start with a quality filtering of the input of the NGS data. The module then proceeds with the annotation to identify the region of interest, like CDR region, and to extract specific features. The module will then cluster the sequence, identify both abundant and rare clusters. The sequence liabilities are quantified, and based on this information, the module will be able to prioritize leads that have optimized information, and they will be delivered as a gene synthesis ready output. So this leads to my conclusion. We are very happy to launch this new service here at PEGS, an end-to-end workflow that will start with screening by sequencing, and it will continue all the way to the recombinant antibody production. So this is a very suited solution for scientists working in antibody discovering, they want to access a high diversity population in their screening. It will allow them to identify the antibodies with high affinity and increase developability because they will have a reduced number of sequence liabilities, helping with further work. The main benefit of doing this work with the GENEWIZ is really that you will have to work with just one partner for your entire workflow. This will ensure security for your samples and for your data that will only be retained by one company. With that, I would like to thank our two partners, OpenAI and Specifica, that have worked hard with the GENEWIZ team to allow to these partnerships and launch, and also to the entire GENEWIZ team that has been working on this project. And if you have any questions, please reach out to me or my colleagues. You can also take a look at our webpage, and if you have additional questions, we can always chat in the break later today. So thank you.
Okay, we have time for about one or two questions. Question right now.
Thanks, Ilaria, for the presentation. It's always good to actually get a new service provider in the market for these things. But my question actually remains the same, which somebody actually asked to Victor as well. Once you get your prioritized list of antibodies, what are the probability of that all of them actually will have a good expression levels? Or are you actually optimizing these sequences later on to make sure that the expression levels are high?
Yes, very good question. GENEWIZ has developed its own codon optimization tool that will allow to optimize codon based on the codon usage frequency in the expression vector of choice for the customer. And it will also help to eliminate certain sequence like repetitive regions or sequences that can impact the DNA or the RNA structure. Overall, using the codon optimization tool that GENEWIZ has, it will implement the expression into whatever vector expression the customers have a choice.
Okay, thank you.
I just have a quick question. So what is the general scalability and adaptability to given clients for this? Are these kind of customizable for certain projects, or are they generalizable for any kind of one size fits all?
So it can be both. We offer this as a workflow, as a package to make it the most easy, convenient to the customer. So GENEWIZ will provide the analysis, and it will be like a straightforward type of approach. But we can also work with our partner to modulate the antibody discovery module to really add any features or information that they might be interested in.
Perfect, thank you. And I think that's all. For time, we're at a refreshment break. Thank you, Ilaria.
Thank
I just want to give you a big round of applause.